I first heard about Terra about 6-7 years ago, and I got an email from Eric, who I didn't know, inviting me to join the scientific advisory board for Terra Ocean. And I am not a marine scientist. I didn't know what this was about. And so I remember, speaking to Pierre Bork, who also was somehow on the email, and he said, do it. The people are fun and the science is interesting. And he was right. And the first advisory board maybe was 6 years ago. And I remember coming back from the meeting and people asked me, well, what are their goals? What are they trying to do? And I said, I don't have a clue. And I don't think it was because I was so jet-lagged at the meeting. I don't think they had a clue at that point. But clearly they eventually got it, and that's why we're here today. And a final thing about, I'm very interested in large-scale science, and I'm curious about organizational structures. Terra Ocean is quite an unusual organizational structure for a large-scale science project. It was a bunch of cats, all going in the same direction. There was no herder really, other than maybe Stephanie, but the herder was a shared vision, and that I think is what brought us here today. And so I'm going to, I was given this title of marine science, genomics in the 21st century, and it's very hard to spend 30 minutes talking about the future. And so I thought what I would do is talk a little bit about the past, a little bit about the present, some of the work going on at Terra, but also work going on in my institute, which has parallels, and then ponder the future. And so really one of the major questions we have, as Homo sapiens is what else is out there. And we can spend a lot of time looking outside the Earth. I think what has been most fruitful and is really demonstrated by Terra is aiming our microscopes towards the Earth, and particularly the wet part of the Earth. And so what Terra is doing, what I'll talk a little bit about, is searching for the boundaries of life on Earth. And I think we're going to, in many ways we're cataloging things, but I think that we're poised to make very fundamental discoveries in addition to cataloging about life. And the reason I'm so optimistic for this is that the role that technology plays in scientific advances, and it's just really leapfrogs us. And we've seen this. And for example, astronomy and astrology, they were one field. There was nothing distinguished them until the telescope came along. And astrology is now something that's done by fortune tellers and tents, and astronomy is what scientists do. And the development of the telescope really gave us a new view of where the Earth fit within the solar system. And there have been a series of technological advances in astronomy that really have, changed our view of where the Earth and our solar system fits within the galaxy and the universe. And so this is where technology is really driving us forward. It's really when we get these new tools. I think the same thing has happened with biology and our view of life. And a precursor to Terra was the journey of Captain Cook, who brought along a scientist, Joseph Banks, and he was both interested in megafauna and megaflora as well as the movement of planets. And in 1768, they took a route not so unlike much of the route that was taken by Terra. They did, in fact, discover some of the places that were visited by Terra. But they also were working with a view of life where the Earth was filled with two forms of life. It was what we could see with our eyes. It were plants and animals. A technological advance occurred, and a Dutchman developed this very primitive microscope and looked at some of the scrapings between his teeth and suddenly realized that there was not just megafauna, there were microscopic fauna and little micrules. And his view was that we had bacteria and archaea. They were distinguished by the fact, bacteria and eukaryotes, and they were distinguished by the fact that eukaryotes had nucleus. And that's what you could see through with a microscope. Along comes very primitive DNA technologies. And Carl Woese, looking at small snippets of RNA sequence, was able to catalog life or microbial life, and drew up the three-domain view of life, bacteria, archaea, and eukaryotes. And so this raises the question, is that all? Are there not other domains to be discovered, or other phyla to expand upon our views of the world? And I think right now we're at this point of a technological advance, and we've just recently entered the massively parallel sequencing era, as well as approaches for looking at uncultured organisms. And so we're, I think, poised to begin not only adding incrementally to what we know, but adding dramatically to what we know. And so I'm going to take you through some recent work from my institute that connects to this. It also connects to Terra. And this is a project that we began. It's called the Microbial Dark Matter Project. And the goal of this project is to begin cataloging uncultured life, to begin looking at the phylogenetic tree and putting genomes at every places that we know exist in the tree. And so this is our view of bacteria and archaea. And these are phylogenetic tree. They're basically around 93 phyla. In dark black are the branches where we know a phyla exists, and we know it exists because a little 16S fingerprint, but we have no genomic information about it, and we know nothing about the organism where it lives. Or we know where it lives, but we know nothing about its lifestyle. And so the goal of this project was to add genomic information to each of these dark phyla. And the approach was using large-scale single-cell sequencing. And basically, it's putting through a sorter individual cells, capturing those individual cells, little microwells, and then amplifying the genomes present in those microwells. And we amplified 3,000 genomes, and then eventually taking an aliquot from those wells, and then choosing which well contains a phyla that doesn't have a sequenced genome, and it's to begin sort of capturing all these uncaptured phyla, and then sequencing these. And so from this project, we generated 200 single-cell genomes. And what we contributed was, in red, are all the phyla that previously did not have sequenced genomes, but now do from this project. And there are still a few that are awaiting capture. Those are the dark black, and we're looking very actively to capture these. Okay, and so insights from these, and I've been involved in lots of genome papers, and usually there's very little information in them. I think Terra Ocean has an enormous amount of information in it, but usually it's just another vertebrate, another mammal with no real stories. And this one really had a lot of stories because it was covering all this phylogenetic space that had never been captured before. One of the nice parts about it, and I think biologists here will appreciate this, is that I'm at an institute, the Berkeley Lab, and it's very much a physics institute. And you can see by the logo, the cyclotrons on the logo, the roof of the cyclotron. And a lot of people studying dark matter there, and the physicists don't give the biologists enough respect. And so I like to thought with this paper that had the word dark matter in the title, that these physicists searching dark matter get to learn a little microbiology. Okay, so one of the insights that really tickled me, and it also leads to how little we know and how the data is available to, or how we've been biased by the cultured world, is that in this data set we had two genomes that had very small genes, that an average length of a gene is about 900 base pairs. In the data set we had two genomes, two gracilobacteria, that had genes of 200 base pairs, these tiny genes. And we spent a lot of time scratching our head because they were so small, they weren't whole proteins. It turned out that it was operating on using a different codon table. UGA in the canonical codon table is a stop codon. What's going on in these two bacteria is that they were using a different code. They were using UGA, they were treating it as an amino acid. And so what we had been looking at was looking at the sequence and annotating every time we saw UGA as a stop, and that was giving us these small gene fragments. When eventually we used a codon table where it is not a stop, but instead is coding for an amino acid, we see normal-sized genes. And so it was these were two organisms were using different, they were not using the canonical code, and quite clearly by aligning the translated product against known proteins, we're able to see that the stop codon was really coding for glycine. And so it had decided, this organism decided at some point to stop using this code that normally is a stop codon and now use it as a coding for an amino acid. And it also raised the issue, is this occurring more often than we previously appreciated? Is it a rare phenomenon or is it a common phenomenon? And this, the issue is the code universal? Do we have one language that all life speaks to translate genetic information, or are there other codes operating out there? And can we look at this from a different window? And we want to look at this in a large-scale environmental way, and this again connects back to Terra, is that instead what we did is we went to metagenomic data. And Terra hasn't added to this, but this is one data set that we have that we're now adding Terra's information to, and that this was looking at metagenomics data, 1,200 different samples collected from around the globe, many of them ocean samples, and we looked through this data specifically for what other codes were operating. And so what we did is we took the data and we translated the data using the canonical code. We also translated it using 3 different possible codes. And possible codes, in this case, we were just looking for stop codons that are now being converted to amino acid codons. And amber, ochre, and opal, or UGA, UAA, UAG, were now being coding for amino acids. And we looked at this massive data set. And what we found is it occurs all the time. And the size of these circles are a couple of percentage of the reads are now using these alternative codon tables. In blue represents where opal is being recoded as a amino acid, and orange is ochre and amber. So there are many organisms out there that are using different codon tables where they have now taken the stop codons and have converted the stop codons into go codons, or coding for an amino acid. And it occurs around the planet, terrestrial, as well as the oceans, as well as the human body. But I think it illustrates what you can do with these metagenomic data sets. You can begin looking at it with different perspectives. We're frequently used to looking at metagenomic data sets and thinking about identifying organisms. We can use it for a data set to begin to tell us basic principles in biology. And looking where this recoding occurred, we can begin to see it's not a random process, but you can see back in time that certain organisms, precursors, certain phyla, made the decision to give up a stop codon and convert it to an amino acid codon. So it's not a random process. So we can see basic principles about how biology was operating billions of years ago. And so in summary, the canonical genetic code is one of the many languages that are used in nature. Looking through metagenomic data provides us a window into that. We have now looked for changes in other codons, sense codons, one amino acid to another, and at a massive scale we can look at the different codon tables that are operating, both for stop codons as well as sense codons. And as I said, it illustrates the power of metagenomic data sets to reveal novel insights into the diversity of life and how it operates. Okay, so that was really an outgrowth of what I called the dark matter project. I'll next describe a quest. And the reason this is a quest is it really has very ambitious goals. And one of the goals are to increase the number of phyla, to see if we can take alternative ways of looking at data sets to increase the diversity of life that we know. But the real goal is to see, is there a 5th domain? Can we really aim our telescope specifically to look for that, to look at data in a different way to see if there are ways to identify another domain? And so one of the ways to do this is to search metagenomic data for outliers. And again, as I said, we have to view metagenomic data as like Twitter feeds, or Google searches. We're frequently wanted whole messages, but there may be insights from the words that are in the messages. People can now predict infections from Google searches and when they were going to occur. The same way we can use metagenomic data like that. And I think with technologies in the future, we're going to be able to use it even much more so. And so there are different ways. This data is very recent, massive scale metagenomic data. And there are new ways of looking at it. And so some of the ways that we're looking at it is nucleotide composition, not worrying so much about genes, but see how
how frequently certain nucleotides are next to each other in combination, getting tetramer or 6 or 7 mer frequencies, and looking if there are outliers. That's what we're specifically looking for tRNA structures, codon usage, and phylogenetic marker genes. And these are ways that we're searching through this data, trying to look for new phyla as well as the possibility of new domains. And so again, I'll describe one of the successes we've had in this area, and it has to do with marker genes. The approach, again, is in this case, to take 5.2 terabases of unassembled sequence data. A lot of this is dirt. The greatest diversity is in dirt, but a lot of it is soil, a lot of it is ocean data sets that we had previously available, and Tara is being added to this. A lot of it is the human body. And we've looked through these data sets just specifically for marker genes, 16S marker genes, and taken out the metagenome. Most of us have looked through 16S marker genes by doing amplification. This is sort of a genomic approach for looking for these particular phylogenetic marker genes. And we've developed trees from this. And in this study, in looking at the 5 terabases of data, we generated 187 intact 16S genes. And then we made a tree of them and looked for outliers that didn't look like anything we'd seen before. And we found several of them. And the most is this one, a very deep branch in bacteria that had less than 82% identity to the nearest 16S sequence. And so it really is definitely a new phyla. One of the questions we had is, why is this phyla not previously been discovered? Because most of the sites that we had looked at, we had done, had amplification, 16S surveys done. We hadn't seen this sequence previously. And so when you do 16S amplification surveys, you're dependent upon these universal primers to amplify the sequence. Well, we found that in this phyla, it didn't match the universal primers. And so it got missed because of that. And so it was a taxonomic blind spot. And we found several other new phyla that had been lost in this taxonomic blind spot. And so we named this new phylum Kryptonia. I was a great fan growing up of Superman. Krypton was his home planet. It's his weakness. We did not name it for Krypton and Superman. We named it for the Greek word, crypto, which means hidden or secret. And so this phyla had been missed because it lacked the sequences required for amplification. Where is it found? And in this study, we looked at 162 unique locations around the planet as well as around the human body. And we looked at 164 unique geothermal, hydrothermal springs. And we found the site exclusively in 4 of them. The exact same phyla, nearly identical sequences found in 2 sites in Asia, 2 sites in North America. We've been able to show that there were viruses at those sites that match them, and those are the only 2 sites that we see them. And so it highlights the diversity of environments that we have, and though all bacteria are everywhere and they're looking for their niches, there are some unique niches that a very small amount of diversity is found there, and that it won't be found everywhere. And maybe if you sequence deeper, you may find that. And so in addition to Kryptonia, we have found 2 other phyla that, again, are greater than 80 or less than 85% matches to anything that's been seen previously. And what does it mean in context? With regard to the periodic table of elements, there are 118 elements. We have 93 bacterial and archaeal phyla. There are now 96. And so we've added 3 more elements to microbial, bacterial, prokaryotic life. And I think there are many, many more out there to be discovered. And so indicates the likelihood of novel life forms awaiting discovery using new approaches. Sequencing has gone through several eras. The human genome was the Sanger era. 454 was this temporary window of massively parallelized sequencing. Illumina has captured much of the space and all the space in much of the Terra Ocean surveys. I think we're about to enter another era. And this may or may not occur, but I think it's likely it will occur. And this is nanopore sequencing. And this really has the possibility of really changing the game. And And this, right now, Oxford Nanopore is one of the companies that has released a product, but I know Roche and Illumina all have nanopore sequencers. They have capabilities that really have the potential to really transform how we do sequence-based science. Right now, we've been working about the past year with these thumb drive, thumb drive size sequencers. You can keep on loading them. They produce read lengths of greater than 200,000 base pairs. They can also detect methylated bases. Previously we're just looking for ATGCs, but you can imagine if there are other bases out there, these sequencers could detect them. You can imagine somewhere in the future, these sequences, it's not a run, it's a time. It's how long you keep the charge on, the current on. And so you really could keep on adding material to them. You can imagine them being in floating devices, not sending terra out on the ocean, but planting a bunch of these devices that are continually generating data, bouncing it off the cloud, and bouncing it to your laboratory. And so, and I think this future is not very far away. There's such an investment in sequencing technology connected to human resequencing that projects like Terra and people interested, non-humans can ride the slipstream of. Basically, the way it works is there's a hole in the membrane. It allows the current to flow through. The current is regulated. It gets a readout of the current, and each nucleotide shows a different impact on the current. And what way it is you have a ratchet protein that binds to one end, and it feeds the sequence into the pore. And what's done is what you're basically reading is a 5 base pair sequence that's in the pore. And so it gets 4 to the 5th different charges that it expects. And so it's able to make a prediction on a base calling based on 5 nucleotides. And it's been out for about a year. It progressively has gotten better. We're getting about 85% accuracy. I know people with the next level instrument are getting in the mid-90s, and so it's really improving. People are getting very long read rounds, and the throughput is going to dramatically increase. So I think this could really disrupt how sequencing is done and could really change the way Terra operates. And the sailboat is very romantic, and collecting samples on it, but you may distribute these by plane throughout the globe and have them continually, add material, sequence it, turn the machine on, turn it off, turn it back on, turn the sequencer on. In fact, part of the business model of Oxford Nanopore is to charge by time. And so you get a chip, and you would pay like you pay for your phone card. And so the world of sequencing we're just at the beginning, and I think it will dramatically impact. And I think Terra Ocean's data will be fantastically valuable far, far into the future, but we may be generating other kinds of sequence data. And imagine if you could get a few hundred KB read, very deep reads. It's not assembling little bits of sequence. And we all realize that sequence is a linear molecule and not just a statistical assembly of 300 base pair sequences. And so why should we care about discovering new life? We want to understand the evolution of life on Earth, its origins. Did it just occur once? Are there radically different life forms out there? I think that we have tools now to begin looking for that in new ways that we hadn't previously. We want to understand microbial communities and how they work and how organisms work with each other, and this is a way. And finally, there are potentially novel biological machinery that we will be able to discover through this. And so where we will next find new life, I think everyone in this room would vote that we have a better chance finding it here. And so I'll stop and answer if there are any questions.
So we have time for several questions. Chris?
Very inspiring. Are to what extent are the new technologies going to be PCR independent, and to what extent are we going towards really single molecule sequencing?
Well, these pore-based there's very little prep
of these pore-based, micropore-based sequencers, nanopore-based sequencers. There's no PCR involved. You know, it's just a naked piece of DNA. The sample prep is getting easier and easier. You're not really building libraries. You're just putting material in. Current is taking it to the pore. And so it could dramatic, you know, I think PCR, I don't think in library creation will play a minimal role in this. And this is, you know, we're not sure whether this technology will scale, but it'd be surprising if it doesn't. There's such investment, you know, in these technologies driven by the personal genome financial driver.
You still have to extract DNA, right? To get extractions of the DNA? Yeah.
Right, you need to get an extraction of the DNA. But they talk about almost putting raw samples in.
Really?
Yeah, and so I think the prep is going to get easier.